Hi guys, this is Mr. Y. Today this will be the third video in the Dissecting Experiments in Real Life series that I created. Again, each of these videos was looking at a real-life biological experiment and the various aspects that make up that experiment and helping you to identify the different parts of them. So this will be the final video and the final experiment we'll talk about, which means I left the most influential experiment for the last. So let's get started. So once again, and for the final time, again, the anatomy that we're going to be looking at with each experiment is dependent upon you knowing several definitions. For instance, what is the independent variable, i.e. that is the thing that we are changing on purpose, or the researcher, I should say, is changing on purpose in their experiment. What is the dependent variable? Again, this is exactly what is being measured or recorded. What is the experimental group? That is the group where the changes are made, the group we're most curious about. That's being compared to the control group. That's the group we just generally use as a basis for comparison. Constants are things that must be held the same between all the various trials that are run. And then finally, the hypothesis, again, for the last time, if you've not watched this hypothesis video that I've linked here into the PowerPoint itself, the link is here as well. It should also be linked in the description. Make sure you do that because, again, we'll be talking about hypothesis using the blended hypothesis format, which combines an if-then will result in and in comparison to setup. So it makes it nice and clear and robust sounding, if nothing else. So make sure you're clear on all of these things. I went through all that stuff with the other experiments as well, so hopefully you're getting a decent feel for this now. So let's get started. Now, one word you're going to have to know for this experiment is the term tide pools, because that's where this experiment takes place. I know many of you, you grow up in certain areas and you don't necessarily get a chance to see coastal regions. A tide pool only occurs where the ocean meets the land, and it's these little sink areas where when the tide rolls in, like high tide rolls in, the ocean can get in here, but then when the tide goes out, these places are kind of isolated. So they're like their own little mini ecosystems if they're large enough, and in many cases they are. People enjoy swimming in tide pools and stuff, but they do have their own little ecosystems at work inside these marine environments underneath the water here. So that's where this experiment takes place. Again, if you look at the actual textbook definition, isolated pockets of seawater found where areas of the ocean meet the land. Here's another picture of a tide pool. Again, you find these all along the, especially the western coast. Not so much along the eastern coast, though in a few places I'm sure you do, but it's usually along the western coast of the U.S. is where we find most tide pools in the continental United States. So the experiment we're talking about today, I've dubbed as the tide pool experiment. Some might know it as the starfish experiment, but it is actually a really big experiment in biology. And what it involved was a young professor at the time by the name of Robert Payne, who designed this experiment, I believe, working in tide pools. I want to say in Washington. It might have been Oregon, but it was somewhere along the northwestern coastal regions. And he wanted to see what would happen if, in understanding how a tide pool's ecosystem work, how its food web worked, this is the food web he was looking at, what would happen if the top predator were removed? Now, in this ecosystem here, this is the food web. Again, you can see these down here are the producers. These are the primary consumers, and then secondary consumers a little bit higher up. And then at the top, you can see the primary predators. In this case is a starfish, a particular type of starfish. In this case, it's called a purple sea star. I'm not going to try to pronounce its genus and species name, but that's the genus right there, Piaster. And for the most part, these were the top echelon of the food chain. And Payne was curious about what would happen if the top predator were removed from the tide pools exclusively. Nothing else, just remove the top predator entirely. So one might expect that, well, if you remove the top predator, then all the other prey species that it directly feeds on, they're all just going to increase. Big whoop. There's just going to be, there's going to be more chitons and limpets and whatever, gooseneck, barnacles. All these things are going to increase. And that would be a very, very, superficial understanding of what actually happened, because what actually happened was once he removed the starfish and he came back many, many times over the course of several years, everything disappeared except for one species. So we'll get to that. The entire ecosystem essentially collapsed on itself. It essentially came down to one species left over after the starfish had been removed. So here's the basic results. Again, this is before starfish removal. This was the ecosystem before he had done anything in the tide pool. After 1 year having purposely removed the starfish, and basically, and you'll see this eventually in the video I show or that I link to, he just went around and he took like a crowbar, something there to pry the starfish up, and he just threw them back into the ocean, away from the tide pool, so they couldn't come back in. And then he would come back every few days, every few weeks, and remove all the other purple starfish that had happened to wander back in. So he just kept the starfish population from getting into this tide pool. He didn't touch any of these others. But after 1 year of removal, you can see the number of species starts to decrease. And initially, he had 16 species in this ecosystem that he had associated within the tide pools he was working on. 16 different species. After 1 year, they were down to only 8 species of the original 16 remaining. So 8 others had disappeared. Within 5 years, only 1 species remained, the mussel, which you can see over here. So yeah, the entire ecosystem basically disappeared, all because Payne had removed the top level predator. That's a rather interesting result. Now, the essence of what he had done took a little while to figure out, but he repeated this experiment with other species. So he found very similar tide pools, and he removed, for instance, say the chitons, and in another tide pool he removed the limpets, and in another tide pool he removed the barnacles. In no other case, though, did their removal actually impact the rest of the ecosystem. It didn't change anything. The number of species was still at 16, as it originally had been. It was only by removing the starfish that we saw this collapse occur down to 1 species. And because of this, Payne dubbed the starfish with a unique term that we now know and take for granted in biology called a keystone. He said the starfish is a keystone species. If something happens to the starfish, the entire ecosystem basically collapses. And that's where the term keystone species comes from. This entire experiment created a whole new field in biology called restoration ecology, because it came to understand how whole ecosystems could fail, how they could end up being decimated by the removal of just 1 species because of the way it's interconnected. And we will get to this in class. But yeah, it's a massive experiment in biology, for one of the simplest experiments you could ever actually do. So here is the video. It's a very short video, like 4 or 5 minutes, showing Payne's experiment. Again, the video is linked right onto the PowerPoint here. I've also linked it here, and I will link it in the description below. But go ahead and take a look and watch over what he did and what essentially happened, because it had humongous repercussions for our understanding of how ecosystems function. Here's a picture of Payne himself in the flesh, and this is like modern times. This is probably back in the 80s, and this is a clip from the video itself where, again, he basically went around and just removed the sea stars. So let's see if we can dissect the basics of how this experiment was working. First, what is the independent variable? What is it that Payne changed on purpose? Well, that was simple. You either had the presence of the purple sea star, the top level predator present, or the absence of it, were the ones where he removed it. And again, he did do other trials where he removed all the other species one at a time in different tide pools to see what would happen. So he had multiple trials going, but the thing we're really changing really focus on changing is the top level predator, the sea star, this guy here, the purple sea star. Is it present or is it missing? What was the dependent variable? Simple. What was being measured and recorded? The number of species that we find in the tide pool as time passes. And again, he was doing these experiments for years and years and years. This was not like a 1 week thing only. He was doing this for several years on end, where he would go in and pull out different species from different tide pools and then just clear them out and see what would happen. Now, What was the experimental group? This is the group where changes were made. Again, you could argue that every one of the tide pools where he removed one species or another species, that was the experimental group. But really, the experimental group we're really curious about is the one where Professor Payne, removed the Piaster species, the sea star. Again, he did this actively for years, going up like every other week to remove sea stars. The control group, the groups we're comparing to, this would be the tide pools where the sea stars were present, or the tide pools that were essentially left entirely alone. What were the constants that had to be kept the same? What did Professor Payne have to make sure was being held the same every single time? Well, you had to make sure that the general size of the tide pools was about the same. So you don't want to compare like a tiny little puddle compared to a huge massive tide pool. That should make sense. Same basic geographic location. Obviously, you don't want to be testing a tide pool in like Oregon compared to a tide pool in say Southern California. All that stuff, just controlling for the geographic location, controls also for like the weather, so storms, the actual timing of the tides, lighting, pollution levels, all that stuff gets controlled just by controlling for the geographic location, because you're all hopefully going to have the same amount of all that stuff no matter what. He had to make sure that you had the same starting species in each case, that all these 16, 17 species existed in each tide pool, or else they wouldn't be a good comparison. And he had to make sure he was using the same amount of time in doing his counting. That is to say, he couldn't count the number of species in a tide pool, say after 1 week in one pool, and then go back 5 weeks later and count this number of species in another pool. No, you have to make sure the counting was done all at the same point in time. So the same time interval gaps between when he did his recordings. All that has. So what would Professor Payne's hypothesis for this experiment look like? Again, we are going to be using the blended hypothesis format. If, then, will result in, and compared to. So if, what is causing what, then, what will you change exactly and how? Will this this will result in what effect will it have and how's it going to be measured, and compared to what is your control or comparison group. So with that in mind, this is how I propose that it be written. If purple sea stars are important as top predators in keeping tide pool ecosystems diverse, that is to say, keeping them diverse with different numbers of species, then by removing all the purple sea stars from one tide pool and leaving them in another, that is to say, that's what we're doing, we're removing the top predators in one tide pool and leaving them present in another, this will result in the number of species declining in the tide pool ecosystem that lacks the purple sea star, so the number of species is expected to decline, and compared to the number of species counted in the tide pool where the purple sea star is left in. And again, I know it sounds all very wordy, but when you read it all together, it should make sense. If the purple sea stars are important as top-level predators in keeping tide pool ecosystems diverse, then by removing all the purple sea stars from one tide pool and leaving them in another, by removing them, this will result in the number of species declining in the tide pool that lacks the purple sea star compared to the number of species counted where the purple sea star is left in. That's a very simple application of the if-then will result in and compared to blended hypothesis format. And I hope that makes sense why I wrote it that way. Again, there's slight variations you can do here and there, but that's the gist of it. Now, here's actually Professor Payne's real results. This is taken from his data. You can see he started his experiment in 1963, and at the time, he had around 16, 17, 18 species in his control and experimental group. His experimental group that he removed the sea stars from actually had a slightly higher counting of species. And then when he came back in 66, you can see, well, actually, he came back in 64 and 65, but you can see the number of species just dramatically drops all the way down to like 1 or 2 with the experimental group without the sea star. And then the control group where the sea star was left alone, the diversity actually went up. It went from like, it looks like maybe 16 up to 20 almost. So yeah, this was a huge revelation that by removing the top predator in an ecosystem, you can cause the entire ecosystem to collapse. It's not that you're going to see the prey species all get huge, because what happens is one of the prey species, in this case it was the mussels, tends to dominate, outcompete the others, and the entire ecosystem becomes destroyed. And while it seems like, okay, big deal, he learned how to destroy an ecosystem, what this really tells you is not how to destroy an ecosystem, but how to restore an ecosystem when we find one that's out of balance. That's what this entire process actually ended up informing biologists on. How to actually take ecosystems that have been decimated and in some cases help bring them back with success. We know this has worked. There have been trials of this basic principle before, where you transplant in a top-level predator and whole ecosystems become revitalized because the predator is there to help make sure no one prey species becomes dominant over the other, and you get more diversity within the ecosystem. We've seen that repeatedly with wolves in Yellowstone that have been transplanted there, and many different places in Africa where ecosystems have been decimated by war. I think I'll show you guys a video of that later on in the year. And then eventually top-level predators get reintroduced and you see diversity of species go up. And so the branch of biology that this influenced is now called restoration ecology. There's whole journals on that, like you can see here, and it's a big, big discovery for how we can actually repair nature. Not how to decimate, but how nature can actually repair itself just by ensuring that top-level predators are present in many cases, especially if they're keystone species. So that was our third experiment. Again, make sure you have watched the video on the hypothesis, how to write a strong hypothesis if you've not done so already, and go back and watch the video concerning this tide pool experiment. Because yeah, there's no underestimating its importance. It was a massive experiment that had huge implications on how we look at preserving nature. All basic approaches to preserving nature now start with understanding this experiment that Professor Payne did. That's it for our video lecture series, guys. Make sure you let me know if you have any questions, and I'll see you in class.